We are next author Dongjae, Kishori, Konwar, Niels, Halson, and Stephen Hallam presenting paper. Authors present here. An automated software tool for processing environmental sequence information using Hadoop.
Hi, I'm David, and I'm working at a bioinformatics lab that does metagenomic that specializes in metagenomics. So today I'll be presenting Koonkie, an automated software tool for processing environmental sequence information with Hadoop. So there are approximately 10 to 22 stars in the universe, and there's approximately 10 to 30 microbes in the universe on planet Earth. And of these 10 to 30, of the small amount that we can sample and sequence, we believe that we only know about 1% of it. So that leaves 99% of those microbes unknown. So we don't know about their genetic material, we don't know about their proteins, and we don't know about their functions. So the study of metagenomics is trying to find out how we can learn about these microbes, because most of these microbes can't be cultured and grown in a lab because they depend on a certain community or an environment. So metagenomics has lots of applications in health, synthetic biology, and ecological monitoring. So in health, you can collect microbes from all parts of your body. So you can collect them from the gut, skin, ear, etc. And in engineering, we also are able to collect microbes to help us build things. So for example, like biofuels or concrete, or new ways to break down petrochemicals. And for ecological monitoring, we use microbes as biomarkers to test for the health of an environment. So for the human microbiome, which is specifically the gut microbes, the microbes in the gut, we can study the microbes in the gut to figure out how they interact with each other and how their interactions affect your health in terms of how you digest nutrients, and vitamins and such. And by finding out what lives in your gut, we're also able to find new ways to find new antibacterial medicines and such. For synthetic biology, using microbes, we were also able to develop the anti-malarial drug. It's called artemisinin. And this was a huge breakthrough because it was able to bring down the cost of anti-malarial medicine from roughly $3 to 20 cents a dose. We can also use microbes in biofuel production, so for example in ethanol, and we can also use them to break down lignin, and produce useful byproducts. And finally, for the environmental monitoring, we use microbes to see how the acidification of the ocean is happening, how certain cycles are working, such as the nitrogen cycle, and we use them to test for oxygen minimum zones and for heavy metal content. So due to next generation sequencing, we've had an explosive growth in the amount of information in terms of genomes we have about microbes. So as you can see, every year it's increasing at an enormous rate. And as a byproduct of that, we also have an enormous amount of protein information that the genes in these genomes would map to. So this becomes a big data problem because in genomics, we don't necessarily know which genes will map to which proteins, and because there's so many of them, and because we have so much data, we need to be able to compute them efficiently, and because both sets of data are increasing exponentially, we have a very large growth in the data we're dealing with. So there are 3 specific tasks that we use in genomics to analyze these data. So we have homology search, the best known example of which is BLAST, which is a method that we use to find the best possible match for a certain query sequence in a database of known genes. We also have a protein family matching, which uses hidden Markov models, where each protein family is represented by a hidden Markov model, where the states are the protein subregions, and we use that to classify proteins into which family it might belong to. And then we also have assembly algorithms, which are ways to take in raw read data and assemble them into longer sequences, which we can then reference again against a known database of genetic information to identify them. So a lot of these big data tasks in genomics have a very similar structure. So what will happen is the query data would be processed into some sort of intermediate form by an algorithm. So in homology search, it'll be processed by the seed and extend algorithm, and in assembly, it'll be processed by the overlap layout consensus algorithm, or through the de Bruijn graph transformation. And then that intermediate byproduct is then constantly referenced against a very large database in order to find the best possible match to identify it. The problem is these very, very large databases and models in genomics, and the amount of computation, it's too large for a single machine, so we tend to distribute it across the cluster. But because every single node in the cluster needs to have a copy of the model or the reference database in order to participate in the computation. So that tends to cause the network to throttle, because these databases can be enormous, and it's a very large obstacle to scaling, because every single additional node to the cluster will have to bring down its own copy of the side data from the distributed file system that supports it. So we chose Hadoop to solve this side data problem. And the reason we chose Hadoop is because Hadoop is very well known. It's very popular, so any improvements will be constantly iterated upon. And also because recently, after version 2, the Yarn Resource Manager was put into Hadoop, and the Yarn Resource Manager is also will be the resource manager for several other future Apache computational platforms, such as Storm, which does stream processing, Tez, which allows you to specialize your directed acyclic graphs for your jobs, and Spark, which allows special in-memory iterative processes. Hadoop is also very fault tolerant, and it's also very easy to program in. It has APIs in Java and Python, and it's far more easier to program in than traditional solutions such as MPI. So there are existing solutions to Hadoop for the side data problem, but they have problems with them. So in a regular Hadoop cluster that runs just Yarn, each worker in a node will just have to naturally bring down its side data from the HDFS by downloading. But the problem is that the side data has a cache timeout. So for very, very long tasks, which these tasks tend to be, the cache may be cleared after each job, which is not optimal because these side data are used over and over and over again. Then there's the distributed cache. There's the distributed cache option, which works quite well, but unfortunately, you still have to bring down your side data from your distributed file system at least once in order to keep it around. Thankfully, though, after Hadoop version 2.3 and beyond, there's short circuiting, which uses Unix domain sockets. And the way short circuiting works is that it allows each node in the cluster to use whatever blocks of HDFS data are stored on that node. The problem with that is that you can't predict how the HDFS will place its blocks, so it's not a very reliable solution, but it's a step in the right direction. So then what is Koonkie, which is our tool? So Koonkie, the name Koonkie is derived from a large elephant used in northeastern India to capture and tame wild elephants. So in the analogy in our case is our improvements to yarn is our Koonkie, and the wild elephants will be the nodes, which we are trying to force to be able to compute more efficiently with the slide data distributions. Okay. So in particular, Koonkie is a set of improvements that we implemented to the yarn resource management system, and it's targeted specifically at removing the communication cost with downloading the side data for each job. And we're hoping that this will allow people to turn any sort of set of computers they have access to into an ad hoc grid or cluster for biological needs. So we implemented this by heavily modifying the distributed cache classes, as well as the yarn localization processes and the localized resource classes. We also used a set of domain sockets, Unix domain socket code. And the way it works is that Koonkie has a small daemon, which will allow each worker, which will scan a set of certain directories and use whatever files are in that directory for the job. So if you already have the data on the node, you don't have to ever bring it down again. So the way we implemented it on our example, which was a AWS EC2 cluster, was that we baked the side data that we needed into an Amazon image, and that means that as soon as we started up our cluster, we'd have everything we needed for processing. Then this is in comparison to normal Hadoop, where everything would have to be brought down from the HDFS, and you don't know if you'll need to bring it down unless you carefully manage the time of your jobs and your cache timeout policies. So we tested Koonkie on a typical metagenomic pipeline. So there are various tasks in the metagenomic pipeline, but we specifically concentrated on BLAST, because BLAST is a large homology search algorithm, which depends on very large databases and very large query sets. So we ran BLAST on AWS cluster running Koonkie with a sample from the Human Microbiome project, which is DNA from microbes in your gut. And in the database we used was RefSeq, which is a non-redundant set of known genetic information, and as of now it's 86 gigabytes. So that would be essentially 86 gigabytes per node in the cluster. So we used M3.2 XLarge instances for our cluster, and these are the statistics for our stool sample. And in the end, we saw that there's a fairly linear decrease with an increase in the number of nodes in the cluster, which is to be expected. But we ran normal Hadoop with the same databases in the same sample, and that actually took 14 hours. 14 hours and 23 minutes, while Koonkie just took roughly 6 hours. So it was roughly 2.5 times increase. And we believe that this is because each node didn't have to wait to bring down all of its side data from the HDFS. So in the future, we intend to improve Koonkie, and as well as try to target more efficient processing for yarn. So in yarn, each container on a node is essentially independent. So if there were several mappers or reducer tasks on a single node, they would each have to bring their own side data in for localization. And that's a redundant amount of information because they could just read from the same database. So we're trying to implement some way for containers to share across the same machine. And for the project, I'd just like to acknowledge the following foundations and the Hallam Lab team members, which helped implement and give support for this project. And the code for the project and all the associated binaries are on the GitHub. So thank you.